Now we are going to plant transformation from the prokaryotic system to the eukaryotic system. Plants are the most routinely transformed species, and these are really hard as compared to the bacterial cells. Bacterial cells and the animal cells, they require the same kind of competence and transformation technique, except that the animal cells can be infected or transfected with some viruses like retroviruses, okay, for incorporation of the foreign DNA, and you need a cell culture for that purpose. They cannot be grown like bacteria. They have specific requirements for the growth of the cells. Specific lines are used for that, and the infection or transfection by the bacteriophage kind of vectors, retrovirus-based vectors, is very common practice in animal cells. However, plant transformation is quite hard, and there are a lot of transformation techniques which has been established for this one, and we will go through all these techniques one by one. And there are two methods, basic methods actually, for transformation of plant systems. One of them is called indirect method, and that indirect method is based on agrobacterium mediated transformation. Okay. We will study that in detail, and later we will go to the other methods, which are mechanical methods or direct methods of transformation. So you will be capable of differentiating between the indirect and direct methods in the coming part of the lecture. So what we will start with is, we will start with indirect method first, that is agrobacterium mediated transformation in plants. Okay, the most common technique, which is routinely used in most of the laboratories, and then we will talk about the direct methods and the specific reasons for using those methods. So I hope you are very much familiar with the structure on the plant. Sometime you can see a tumor-like structure on the shoot, and this is due to the agrobacterium infection. Okay. So this is a natural process. The bacteria, the agrobacterium, lives in this callus-like, tumor-like structure, and it gets its nutrition from the plant while giving specific benefits to the plant as well. Okay, so if we look into this bacterium, it has got a 200 kilobase plasmid. And this is the plasmid which is responsible for every communication between the plant cell and the bacterial cell. Okay, so this 200 kilobase plasmid is a wild type plasmid. And if you remember from our previous lectures, that the plasmids, which are very big in size, this is very big, as compared to 2 or 3 kilobase big vector that we use for transformation, general purpose transformation in the laboratory. Okay, so you should understand immediately, if this is a big plasmid, it might be having an origin of replication which is very, very low copy number. Okay, and that's a true fact. And all these large plasmids, they have origin of replication which is very slow and keeps the plasmid copy number at a very low level. Now, you also know that we don't use the wild type bacteria or the wild type plasmids in the laboratory. They are all synthetic. The bacteria have genetic modifications, and the plasmids or vectors, they have only the minimum number of required genes to carry out their functions. All the other genes are removed. So this is a key example for explaining all that. The Ti plasmid, which is 200 kb long, it contains a number of 155 genes. All of these genes they are not required for plant transformation. So in laboratory, the size of the Ti plasmid that we use for plant transformation is around 10 kb. So 10 kb is 1/20th of the size of the wild plasmid. Okay, it's very, very small. And then what we do is that if we look at the structure of this wild type Ti plasmid, what are the things of our interest? Okay, the things of our interest is the tDNA. What is tDNA? And this tDNA is actually a single-stranded DNA, which is shown here with a single line just below the double-stranded DNA between the left border and the right borders. Okay, so this is a single-stranded DNA fragment between the left border and the right border, which is produced by this region, which is coding for auxins, cytokinins, and opines. Okay. So this single-stranded DNA coded by all these three different types of genes for specific hormones and the substrates, this single-stranded DNA is transferred from the bacterial cell into the plant cell. One cell produces only one DNA strand. It means one cell produces one tDNA. And that tDNA moves from the Agrobacterium cell into the plant cell, and then it makes its way to nucleus, where it gets double-stranded and is integrated into the plant genome. This is the simplest transformation process between Agrobacterium and the plant cell. We are therefore interested in this region, the region between right and left borders. The other things that we are interested is that the bacterium can utilize some nutrition from the plant cell as well. So it has opine catabolism related genes on it. Obviously, it has to replicate independently. So origin of replication is there, but it's a very low copy number, origin of replication. And then, because the Agrobacterium has to infect the plant cell must be capable of getting infected. And the bacteria, that is the Agrobacterium, must be capable of having the virulence in the strain that should make it capable of infecting the plant cell. And this virulence is determined by a set of different genes. So the Ti plasmid has got about a 40 kb DNA region, which is involved in coding the virulence genes, which trigger the infection of Agrobacterium to plant cell. So it means that we essentially need in a synthetic plasmid some of the genes which are required in infection, in replication, okay, of this plasmid, and then we also need some genes for to act as genetic markers like antibiotic resistance genes, okay. So this part is very important, the virulence region. The origin of replication is very important. We are not interested in the auxins or cytokinins or opine genes. So what we do is that we remove all this part from the plasmid, which comprises of auxin, cytokinin, and opine genes. And we also remove opine catabolism region because we are not interested that the bacterial cell should survive on the plant cell. We are just selfish. We want that Agrobacterium should interact with the plant cell and produce the tDNA. And that tDNA should move to the plant cell and get integrated into the host genome. This is the requirement of a molecular biologist or the person who is expert in recombinant DNA technology. So by removing the auxins, cytokinins, and opines, which consist of about 20 kilobase of the DNA region, okay, so we can put the DNA of our choice in there, because this is the natural tendency of the plasmid to produce a tDNA, because it has got left borders and right borders and the virulence genes. And I will show you the mechanism that how the single-stranded DNA is produced. Okay, but if we replace auxins, cytokinins, and opine with the cassets, the gene cassets of our interest, one of the gene cassette, it should be a genetic marker, okay, that carries the antibiotic resistance gene, so that we can select our plant cells which are transformed with Ti plasmid on antibiotic selection medium. So one of the cassette in this region is always a marker gene, okay? And then because this is quite a big region, 20 kilobase, our cassette for a genetic marker can be 3 kb or 4 kb. We still have the opportunity to clone 16 kilobase big fragment with this one. Okay. Even if we use 4 kb for one cassette, we can clone 3 or 4 different cassettes in a single Ti plasmid, okay, producing a 20 kb tDNA that can be moved. So it means that we can use this region to clone more than one gene. Okay, multiple number of genes can be incorporated in this region and can be transformed to the plant cell. Now, if we look at the basic features of this plasmid, other than what we have been talking about, okay, there are 2 strains of Agrobacterium. One is known as octopine strains, which actually utilizes octopine for its survival. And the other type of Agrobacterium is called nopalin strains. And these strains, they utilize nopalin for their nutritional resources. 2 strains living on 2 different nutrition resources. The octopine strains, they have a Ti plasmid, which is 200 kilobase big. But what it has got, it produces 2 different tDNAs. The diagram that I was showing to you was producing a single tDNA. So this is a nopalin type of Agrobacterium strain that has got this plasmid. This is called nopalin type Ti plasmid. The octopine type, it bakes 2 different tDNA. It means that it has 2 different left borders and 2 different right borders. One pair, left and right border, is covering a region of 14 kb, and the other left and right border is covering a region of 7 kb. So 2 separate tDNA molecules are produced. One is 14 kb and the other is 7 kb. The nopalin strain, however, produces only one tDNA, which is 20 kb. And you saw it in the previous diagram. Okay. The one which is of our interest, we don't divide the genes into different parts. So we want to transfer a single tDNA into a plant genome. So we prefer to use nopalin strain in the laboratory. Okay, the most popular Ti plasmids that are used in the laboratory, they are based on nopalin strain, which produces a single tDNA, which may be as big as 20 kilobase. It can be smaller than that. It depends on the kind of genes that you clone between left border and right border. And I already mentioned the size is 200 kilobase And this is the plasmid which is responsible for tumor formation, which is known as crown gall formation. It contains one or more tDNA regions depending on the bacterial strain, which are integrated in the genome of plants. Whether tDNAs are produced, both of them, they go to the genome. Okay. If you're using Nopalin strain, it produces one tDNA, which gets incorporated into the genome. So, the Vir region, which is 40 kb, it contains 8 to 11 different virulence genes. Okay, we will study these genes shortly, what they are doing in the formation of tDNA, okay, and the process of that. Then we already know that it has an origin of replication, which is low copy number. And this is also having a gene which is capable of conjugative transfer of the plasmid from one bacterial cell to another bacterial cell. Okay, so the Agrobacterium strains which don't carry Ti plasmid, they can acquire them by conjugal transfer from the bacterial strain which has got that. And then the genes capable of catabolism of opines at the nutrition resources. Now, as I mentioned that the 200 kb plasmid has 155 open reading frames, means the Ti plasmid, which is the wild type, it has 155 different genes which are required by the bacterium for different purposes. But we are not interested in all the 155 genes. The 5 major components of the Ti plasmid, they are the T region where we clone our genes between the left and right border by substituting the genes for hormones, okay, the cytokinins, the opines, okay. These region is removed and we use that tDNA region for cloning the gene of our interest. The virulence genes, these are the genes which are responsible for infecting the plant cell and also producing the tDNA. And then the replication region, that is origin of replication required for the initiation of replication of the bacterial Ti plasmid. The TRA and TRB loci, which we already mentioned actually, which are involved in conjugal transfer of Ti plasmid from one bacterial strain to another bacterial strain of Agrobacterium. And then the genes that are capable of uptake and catabolism of opines. These are the characteristic features of the Ti plasmid. And this slide is just to tell you, just to show you the different color bars on this map of the Ti plasmid. They indicate different types of genes. Okay, the virulence genes are shown mostly in red. The conjugal transfer genes, they are shown in brown. The opine utilization genes, they are shown in blue. And the replication associated gene, they are shown with yellowish, greenish yellow color, apple color. Okay. So the ones which we need to develop a Ti plasmid that can be used for plant transformation, they are the virulence gene and the replication associated genes. None of these genes which are covered by blue or brown color, they are not actually utilized at all in the synthetic plasmid. I don't want you to remember this slide. I don't want, just want you to remember that this plasmid, the wild type plasmid, has got 155 genes. So what happens actually with these Ti plasmids is that during infection, an octapin type plasmid transfers 2 fragments because it produces 2 DNA. while the Nopalin type Ti plasmid, it transfers one tDNA, which is 20 kilobase in size. And the characteristic features where these tDNAs are originated, they are originated between the left border and right border sequences, and these left and right border sequences, they have a 20 base pair direct repeats, which are known as border sequences. So these are direct repeats. It means, for example, ATCG, ATCG. This is direct. If you invert the second one, it becomes inverted repeat. Okay? So direct repeat means the same sequence running at one end of the tDNA and the same sequence running at the other end of the tDNA. And this is unique. This is 20 base pair long, and these are called border sequences. The sequence on the left side of the tDNA is known as left border, and the region at the other end of the tDNA is called right border sequence. Okay? So these are 25 base pair long border sequences, which are direct repeats. It does not mean that the tDNA border is 25 base pair. No, it is quite long, like 200 or 250 base pair. But the one which is associated with tDNA is the left border and right border, which are direct repeats. Octoprin Ti plasmid often contain near right border another cis-acting element. I hope you remember from my lecture on the regulatory regions, I mentioned cis-acting and trans-acting. Cis-acting are the DNA sequences which are on the same chromosome and they are quite close to the regulatory region. Okay? So this region, left border and right border, you may consider it to be as a regulatory region, okay? And very close to the right border of the tDNA is an other sequence which is known as overdrive. Overdrive means it accelerates the production of tDNA. But this is only present in octopi, not in the Nopalin type of bacterial strains. Nopalin type, they don't have an overdrive. But when you are designing a synthetic Ti plasmid, you can pick this sequence of overdrive and you can clone it in the Nopalin type plasmid so that you can make your plasmid more efficient in the formation of tDNA that moves from bacterial cell to the plant cell. Okay, so if you ever think of overdrive sequence, it is present on the Ti plasmid from the Nopalin type of strains. And it accelerates the formation of tDNA from the Ti plasmid. That is quite simple. Another possible overdrive element is present near right border of tRDNA. That was tLDNA. What means tLDNA and tR? It means we are talking about Nopalin strain, which produces two tDNA molecules. One is known as TL and the other is known as TR. So the TL tDNA borders, they contain overdrive, and TR, they are also expected to have an overdrive sequence, but their function is not very clearly known so far. So only the overdrive sequence, which is associated with TL, part of the tDNA, is used for some Ti plasmid vectors for efficient development of the tDNA. Now we are talking about the direct repeats of the left border and right border at the boundaries of the tDNA. Okay. So if you look at the sequence comparison between the left border and right border, you see that there are some nucleotides which are identical to the two borders, like this region, starting from GA, GG, AT, AT, AT. Okay, this is quite identical kind of sequence. Then there is a match here at G, and there's T, and an A here. But all these X's, they represent N's, nucleotides. They may be any nucleotide. It means that there is some variation between left border and right border sequences. But they exist in the direct repeat order. It means this is always at the 5' end to 3' end at one border, at right border, and the left border, it is exactly in the sequence. And because most of the sequences they match, so they are known as direct repeats of the similar sequences, but they have some differences. Obviously, you can see it from this slide. And overdrive is present in octopene type of Ti plasmids. And you can pick that sequence and you can clone it in a nopalin type plasmid to make this tDNA efficient. Okay? So this is some of the information that you essentially need to understand the Ti plasmid. Then this is what actually, if I ask you now that what does it represent? It represents two different tDNAs. And which strain of Agrobacterium produces it? It's obviously nopalin type. It produces two different. One is called TLDNA, the other is TRDNA. It has two left borders, two right borders for two separate types of tDNA molecules. So what happens is that when this plasmid is present in the bacterial cell, what do the virulence genes do to produce the tDNA? And for the infection of the plant cell, what is the process? This is now I'm going to elaborate for some time. Now what happens is that virulence genes, they have actually one important virulence gene which is known as VIRD2. There are many virulence genes, like I said, the slide I showed, 8 to 11. And out of those 8 to 11 genes, one of them is VIRD2. So this gene is involved in the formation of tDNA. And how it does is, you know that a plasmid DNA is a double-stranded DNA molecule. And the Ti plasmid has got a left border and a right border if we are using nopalin type of Ti plasmid. Okay. So this VIRD2 protein, it produces a nick in the single strand, one of the strand, the double-stranded DNA between left border and right border, which is about 4 nucleotides from the left border. And then the process of tDNA formation starts. The bottom strands are recovered as single-stranded form referred to as T-strand or tDNA. I will show you the process in a while. So this is the actual process. So if you know a left border and a right border now, you know that there were some genes here in the wild type, octopene, cytokinin, auxins. But in the synthetic plasmid, we replaced all those genes with the genes of our interest. So the genes of our interest, they lie between left border and right border, they are here. And we have taken the overdrive sequence from the octopene type strain and put it close to the right border side so that it can initiate the formation of tDNA very efficiently. Okay. So how it does is, I said VIRD2 is one of the protein which is responsible for producing a nick. I hope you can see this nick where the cursor is there. Okay, one of the strands between the right border and left border is nicked by Vir-D2. And the overdrive sequence binds to another virulence gene which is known as Vir-C. The Vir-C interacts with Vir-D2 in the maintenance of efficiency for the formation of tDNA. Okay, it doesn't, even if you remove this, still the tDNA will be made by Vir-D2, does not matter. But Vir-C efficiently performs this function with the help of Vir-D2. That's only a protein-protein interaction. Okay, because the basic function is by Vir-D2, which produces a nick here, and then which are the proteins which separate the two DNA strands? They're the helicases. The helicase proteins, they come in, and after the nick, they start separating these two strands. Okay, so these two strands, they start getting apart from each other. So this strand which was nicked here is down here, separated from the second strand. And when the strand is separated by the helicases, there is another virulence protein which starts binding to this single-stranded DNA to make it capable of movement from the bacterial cell to the animal cell. And this protein is known as Vir-E. So, after cutting this, after producing a nick, the Vir-D2 protein is separated from the tDNA, which is generated here. The Vir-E starts binding to that DNA, and the helicases keep on unwinding this single-stranded DNA from the rest of the single-stranded DNA. And then, when the helicases reach here towards the left border, another protein molecule of Vir-D comes in and produces a nick, 4 nucleotide inside the left border and the fragment that you have inserted here. Okay, so this tDNA contains 4 nucleotides from left border and 4 nucleotides at the end from right border, and all the single strand DNA gets separated after the second nick by Vir-D2 here. Okay, just 4 nucleotides inside the left border, producing a single stranded tDNA molecule. And this is what is transferred from Agrobacterium into the plant cell. This is all the virulence genes too here in the transfer of tDNA. Okay, some of the proteins, they take this Vir-E bound single-stranded DNA, which is tDNA, to the plant cell. We will talk about those a little bit afterwards, but I hope in the simplest form, we are very much clear that the Vir-D2 produces a nick here. The helicases, they detach the strand from the other strand, okay, and then the Vir-D2 again produces a nick here and separates the single-stranded DNA fragment from the DNA. Obviously, this red arrow shows that the DNA polymerases come in and again make it double-stranded. So the plasmid remains double-stranded after releasing this tDNA. The main function here is by Vir-D2, and as soon as the single-stranded DNA is produced, the Vir-E surrounds this single-stranded DNA and tightly bonds to it. And this tDNA is not now only the DNA, it is the DNA protein complex, only a single protein, where E is binding a single-stranded tDNA, which is generated by Vir-D2 cleavage. Okay, so the second cleavage, By VIRD2 here releases this tDNA, which is bound to the VIR-E proteins, and this is the complex which is transferred to plant cell. And in the nucleus of the plant cell, it gets double-stranded, and then that double-stranded DNA gets integrated into the plant chromosome at a random place. Every time the tDNA goes to the nucleus, it can be integrated in any of the chromosome at any of the place. So it's a random integration. You never know where this double-stranded DNA is going to be integrated into the plant genome. But I am quite sure that you are not now familiar that how a single-stranded tDNA molecule is produced by the Ti plasmid. 3 proteins, VIRD2, helicases, and then VIR-E. 3 of the genes. They have been utilized from virulence genes here. This is the formation of tDNA. This is not the process of infection. Infection is carried out by other virulence genes. What is infection? Infection is the infection or the attack of the Agrobacterium on the plant cell to get that plant cell infected with that. So there are other genes which I will mention shortly. They are not too much. Okay, so there are 8 to 11 genes, and some of them they are important, and we will talk about the important genes only, not all of them. Okay, so the virulence region, it contains some of the very important VIR genes, VIR-A, VIR-B, VIR-C, and I hope you know that VIR-A and VIR-B we have not talked about. We have talked about VIR-C. Okay? VIR-C interacts with the overdrive sequence to increase the efficiency of VIRD2 for producing the NIC on the Ti plasmid for generation of a single-stranded tDNA. VIRD, and out of these, VIRD1 and VIRD2, they are associated in the same function, but the cut is actually produced by VIRD2. So we know about VIRD. We will talk about VIR-E, VIR-G, VIR-F, and VIR-H, 4 more virulence genes later on. But at this point, you must remember that these VIR genes, they have VIR-D, and VIR-D produces 2 different types of VIR-D proteins, VIR-D1 and VIR-D2. VIR-D2 is involved in producing the cut, while the association of VIR-D1 actually improves the efficiency of VIR-D2, but it is not essentially required for producing the cut, so VIR-D2 alone can do it. Now, the virulence functions are transcriptionally regulated. And this is the actual process of infection. And after talking about this slide, I will stop the lecture. Okay. So the virulence functions are transcriptionally regulated by 2-component gene regulatory systems. Okay, how the transcription of virulence gene starts. This is the basic purpose or meaning of this sentence. That how the virulence genes become active. Okay, they get active due to 2 conditions. One is that we know that the bacteria, they are adhering to our skin, but they are not doing any harm to us. They cannot do any harm until and unless we get a cut on the skin. And that cut gives that bacteria entry to the blood cells and the tissues which are broken down there. Similarly, in the plants, whenever there is a cut, there is a damage, there is a release of some compounds. So damage is one thing that is essential for agrobacterium infection. Upon damage to a plant cell, I mentioned that plants, they have big vacuoles, which are their storehouses. And plant keeps all the phenolic compounds, the waste compounds, into those vacuoles to protect its own metabolic activities. And the pH of the vacuole is very acidic, like 4.5 or 5. So when there is a damage, a vacuole gets broken down, releasing all those phenolic compounds and resulting into an acidic pH in the environment of that damage. These are the two triggering events for the infection of Agrobacterium. That is, decrease in pH and the release of nasty compounds like phenolic compounds. And one of those compounds is acetosyngion. The acidic pH, the presence of acetosyngion are the two chemical factors which trigger the expression of virulence genes. And how it happens? How do those trigger that stimulation of the virulence gene expression? We will start it in the next lecture.